Hey everyone! So in today's lecture we are going to be looking at plant breeding, right? We talked a little bit about this already in the previous lecture. I started out by showing you some different pictures of ancestral or wild varieties of our modern cultivated plants. You saw there were really major changes between those wild varieties and the varieties that we plant nowadays, right? Our modern cultivars. And we achieved those for the most part through most of human history. We achieved those differences through this process of breeding. And that is what we are going to be discussing today. So, to reiterate, breeding is selecting a female and male parent with unique and desirable traits and crossing them to produce offspring with desirable traits. So, as we learned, breeding is sexual reproduction, right? And as we learned in the last lecture, most of our organisms, right, they are diploid, right? So within a cell, within the nucleus of the cell, there's all the chromosomes. And diploid means that there are two copies of each chromosome. Diploid. And it's sometimes referred to as 2N, right? And that's the majority of organisms, although there are some that are triploid or more, right? They have more copies. So because of this, because you have two copies of each chromosome, you have two versions of each gene, right? Organisms have two versions of each gene, and we refer to those as alleles. Now, you could be either, for any given gene, an organism could be homozygous, and that means that the alleles are the same. So, same alleles, or it could be heterozygous, and that the alleles are different. And we could express that real easily. We could show that, say we're looking at a gene for flower color, right? And this B, capital B, is going to code for purple flowers. Lowercase is going to code for white. Right, we can have an organism whose genotype is homozygous for purple, homozygous for white, or heterozygous. Right, we also talked about dominant and recessive alleles. Right, and our dominant alleles are expressed if they are in the genotype, essentially, right? So if it's a homozygous dominant, which we see here, and typically for notation, a capital letter is going to code or stand for the dominant allele, right? So this would be homozygous dominant. That's going to have purple flowers. But even in this heterozygous condition, these will have purple flowers because that dominant allele is present, right? And only when we have a homozygous recessive that we have right here will we get white flowers. Okay. And then, again, I said this is breeding is sexual reproduction, right? So what is happening is that we are combining sex cells in this process, ultimately. And to get sex cells, we have this process called meiosis. So as a simplification, we start here. We just have our two copies of our one chromosome, and it's going to break down. There's these initially some replication. Eventually, it breaks down into sex cells that only have one copy of each chromosome. So as a result, one copy of each gene or one allele, right? And this occurs for the male parent. It happens with the female, and ultimately these will, say we have a female sex cell over here, so this would be male, this would be sperm, this would be an egg. They're going to combine, and from it we will get a normal diploid organism that is going to develop into a new plant. right? But this process of meiosis, of forming these sex cells, and then the combination of them serves to mix up genes, right? Mix up genes combinations because it's random which one of these alleles is going to get passed down and ultimately form the new organism. And so we get random alleles that are getting passed down, and then we get half and half. So this new organism is a combination of half the genes from its father and half the genes from its mother. So all of this works together to increase or increase gene combinations of the offspring. And there are good reasons for this. There are beneficial reasons for this, as we talked about in the last lecture, right? One of the main ones being is this increased genetic diversity, or these gene combinations can help protect offspring from really pathogens or parasites that could infect them. But if we are talking about breeding, where we want to maintain certain traits, this could be a drawback, right? Because we may have a gene combination for this one parent up here that we want. Maybe it's a certain flower color. But given the random chance and the mixing up of genes that occurs in sexual reproduction, the offspring might not all share that same flower color as we want that the parents have, or one parent has. And we're going to look now at how that works a little bit, kind of an easy example. And I'm going to show you a tool that you can use, and I think I have some space down here. So a tool that we use to track heredity, a simple heredity, is referred to as a Punnett Square. P-U-N-N-E-T-T? Not entirely sure about the spelling of that, so double check that. I think that's correct. But a Punnett Square. And a Punnett Square is a really simple tool that we can use to track genotypes of both our adults and what we expect the offspring are going to be. And since we know the genotype, we can expect what the phenotype is going to be, so the actual observed traits. So again, we're going to focus here on flower color. Right, and I'm going to draw a line here to try and give us some space. So we're going to go through some hypotheticals right now, right? And we are going to cross a homozygous dominant plant with, we'll do it real easy. We're going to start real easy with another homozygous dominant plant, right? So how you use a Punnett Square is you take each potential allele that could be passed down. So you start with your one parent, and you see we have two potential alleles here. And you're going to write them over on the side here, and we're going to draw a square. Right, so you got your two potential alleles over here, and then you take your other parent, and you write down its two potential alleles. So in this case, they're both capital B's. And then you start in each box, and you bring down the alleles that would fall into that box. So you bring down this capital B, fills in that one for there. That capital B goes there. This is all going to be very simple. Capital B, capital B, capital B, capital B. Right? They're all going to be the same. Right? So in this case, all of the offspring will have the same genotype as the parents, and they will all have purple flowers. Really easy. Right? And we can do the same thing if we're crossing two homozygous recessives, right? We set up our Punnett Square. We know for this parent, we bring down each allele. This one, each allele. We bring them down to fill in the boxes. Right? They're all going to be the same. And here, all of our offspring are going to have the same genotype. They are going to be code for, they're all going to have white flowers. Right? Pretty simple. So let's look at some others now, right? What if we cross a homozygous dominant, so that has purple flowers, with a homozygous recessive, a plant that has white flowers? Well, we set up our Punnett Square. We bring down the alleles for this guy. We'll bring them over here, so we can, you could either pass down a big B or another big B. This one is definitely going to pass down a little b or another little b. And then we fill in our boxes. So we bring this over. There's a B there. We bring that one down. Little b, big B, little b, big B, little b, big B, little b. All right. So in this situation, all of our offspring, again, are all going to have the same genotype, and they are all going to have the same color, right? They have these big B's, so they're all going to be heterozygous. They're all going to be purple, but their genotypes are going to be different than the genotypes of the parents, right? And that's kind of important, because then if we cross them together, say we cross two heterozygotes together, now we're going to start to see some changes in our offspring. Right, so we'll take this guy first, so he can either pass down a big B or a little b, and same with this other one. And then now, as we start filling in our boxes, bringing down our alleles, we see that we're starting to get different gene combinations. So a cross of two heterozygotes is going to give us offspring that have genotypes in the ratio of 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. Right? And thus our offspring will have a phenotypic ratio. Right, so here we have all purple flowers, but now we cross these two purple flowers together, and we're going to get offspring in the ratio of 3 purple, because again, our heterozygotes, these guys are going to be purple because that big capital B is dominant, to 1 white. All right, so now we have, we're starting to see different phenotypes. So if we look at this, and if we look at these kind of basic, easy examples, we can start to understand that if we are trying to maintain traits, right, we want to pass down traits, we want to create a population that breeds true, so the offspring are going to have the exact same traits as the parents, we ideally want to have a lot of our genes be homozygous, right? Because if we have these conditions here and here, we know that if we cross these together, the offspring are all going to have the exact same genotype and consequently the exact same phenotype or the exact same traits. If we start having crossing plants that are very heterozygous for a lot of their traits, right? So it's this condition where we have different alleles, their resulting offspring are not going to breed true. They are going to have different genotypes and consequently different phenotypes or different traits that we see, right? So that's one of the things that occurs, that has occurred in history as we've bred plants. We generally go from a situation where we have lots of genetic diversity and then we reduce that, right? So we start here in the wild, we may have lots of genetic diversity, and then cultivated, we'll have fewer, we'll just say less genetic diversity, more homozygosity, if that is a word. I think it is. All right. So that's what's happening. As we've bred plants through generations, we essentially are removing certain alleles from the population. And what that does, it creates more homozygous plants, right, with these gene combinations right here. And we know when they get crossed with other homozygous plants, the resulting offspring are going to have the exact same genes and they're going to have the exact same traits. Now, this is a good thing in many ways, right? It ensures that the plants that we are producing are going to maintain those traits that we have found desirable, whether it is some of those things I talked about, right? Bigger fruit, sweeter fruit, more edible flesh of the fruit, early maturity, loss of seed dormancy, etc. Right? So all of those have been bred, and now we have a condition where these plants are more homozygous for all of their traits, and we know those traits are going to get passed down. The downside of this is when you have low genetic diversity, your population is more susceptible to disease or changes in environment, right? It's this genetic diversity that lets our organisms adapt to changing conditions. So if you don't have this genetic diversity, your organism or your population really is not going to be able to quickly adapt to whether it is changes in the environment or pathogens that could potentially attack and wipe out populations, right? So that is the danger of these long-term breeding programs. And it's why, in the article that I'm having you read for this week, it's why scientists for the past couple of years have been looking and trying to collect wild or ancestral varieties of some of our crop species, because they're trying to get back some of these lost genes that have been bred out, with the hope that they can incorporate them back into some of our cultivated varieties to improve their genetic diversity and improve their resilience to whether it's pathogens or now a changing environment and changing climate that we're seeing. So, and actually, I can give you a quick example of the dangers of this low genetic diversity, and that's our bananas. Right, so all of our bananas are propagated vegetatively. And as we talked about, that means that they're all clones of each other, right? They don't go through this sexual reproduction process. So about 100 years ago, the main variety of banana that was propagated was the Gros Michel. And it was the main propagated variety. And then there was a fungus, Panama disease, that developed or was able to attack these banana plants. And because all of these banana plants were all genetically identical, that fungus was able to infect all of them. And the Gros Michel banana was essentially wiped out, right? And there was fear that the commercial banana production was going to be gone. Luckily, there was another variety of banana, the Cavendish banana, that was resistant at least to that strain of Panama disease, and we were able to plant that, and that has taken over. So now all the bananas that you eat are Cavendish variety bananas, right? If you went back before the 1950s, I believe, they all would have been these Gros Michel, a completely different type of banana. The unfortunate thing is, again, these Cavendish bananas, they're all propagated vegetatively. They all have the same genes, and there is another strain of Panama disease that is infecting the Cavendish bananas. So there is a chance that unless we can find some sort of resistance or find a new strain of banana, all of those bananas, the Cavendish bananas, are going to go the way of the Gros Michel, and we will lose out, and we won't have a large commercial banana production anymore, right? So enjoy your bananas now while you can. Okay, so I think that gives us a good overview here of some of the genetics behind breeding. Last thing I want to do is show you that we can do this with more than one gene, right? So there we were looking at one gene. We said it was flower color, right? Hypothetical flower color. We can do a Punnett Square with two genes, right? And we'll say we're going to have another gene now that looks at leaf shape. We'll have two versions here. And that's going to look at leaf shape. And we'll say this L is going to be a lobed leaf. Oops, lobed leaf. And that L is going to be an entire leaf, so no lobes. Right, so lobed leaf would be something that looks like that, and an entire leaf is just going to look like that. So, and again, capital letters are dominant, so lobed leaf would be dominant. Really, this is just to show you how we can do a larger Punnett square. Two genes is about the max that you're going to do with Punnett squares. After that, it gets way too complicated. But for simple stuff, like simple traits that are affected by one or two genes, this is the way to go. So, we can have a potential cross, and I'll just make it up. So say we have this plant is heterozygous for flower color, and it is homozygous for lobed leaves, and we're crossing that with a plant that is homozygous recessive, and we'll say homozygous recessive for everything, right? Okay, here's our situation now. So how do we set up a Punnett square for this? Well, it's very simple. What we do is we're going to take each potential allele combination that could be passed down, right? So if we start here, we could have for our first gene here, we can have this capital B allele, and that could get passed down with a capital L allele for over here, and it can also get passed down with that allele, right? In that case, they're both the same. Then we could have a situation where our other allele gets passed down, lowercase b, and it gets passed down with that one, or it gets passed down with that one, right? So we have 4 options of allele combinations that could get passed down. Any of these could end up in if this is the male, a sperm cell, and those could get passed down to the next generation. And we do the same thing over here. In this case, they're all going to be the same. So we have this allele can get passed down with that one, or that one, and this allele can get passed down with that one, or that one, right? Again, we got our 4 combinations. And then we do a larger Punnett square. That is a little wonky, but you get the idea. Right, and we set up this situation where each of our allele combinations is going to go in a spot. So we'll start with these guys over here. Whoops, didn't need to do that. Right, so we write each potential combination on the side, and then each potential combination up here. And then we do the exact same thing that we do for all of them, right? So we bring this one over, so that's a big B, this one down, little b, this one over, big L, this one down, little l, big B, big L, little l, little b, big L, little b, big L, little. And you guys get the idea, right? And you can do this for all of them and fill them all in, and that will give you the expected ratio of genotypes and phenotypes that you should expect for your next generation, right? So have a handle on this, because you're going to have to do one of these Punnett squares in your breeding lab. Okay, so let's go back now. Okay, so now that we got a little bit of a handle on the genetic basis of breeding, we're now going to take a look at some of the practical applications. Right, and one of the first things that we need to know if we're going to establish some sort of breeding system is we need to know the way in which our plants are reproducing, particularly in regards to how they pollinate, right? Whether they self-pollinate, they cross-pollinate, or they do something called apomixis, which is less common but does play a small role in some plants. So, self-pollination, this is where a plant can fertilize itself, right? So pollen from the anther lands on the stigma of the same flower or the stigma of a different flower on the same plant. And the anther is part of the stamen, which is the male reproductive part of the flower. Stigma is part of the pistil, which is the female reproductive part of the flower. So in self-pollination, these plants can fertilize themselves. Now, the degree of self-pollination will vary among species. It's more common in our cereal grains, so barley, oats, wheat, and rice, and our legumes. So these could be very highly self-pollinated, where in some of these species or varieties we see less than 4% cross-pollination. So they're primarily self-pollinated. In cotton, pepper, tomatoes, they will also self-pollinate, and they're also self-fertile, but they also cross-pollinate at a greater percentage, right? So not as much. They cross-pollinate more than our cereal grains do. Now, most of our woody species will not self-pollinate. Cross-pollination. So this is where the transfer pollen from the flower of one plant to the transfer pollen from the flower of one plant to the flower of another plant, right? So here, our plants will not self-pollinate. We need to get the pollen from a different plant to pollinate and fertilize our flowers. So this is common in nature. And it is this cross-pollination that is going to increase genetic diversity of wild populations. And we already talked about genetic diversity and how important it is for evolutionary adaptation and making resilient populations. So it's really good in nature. It is less good if you are trying to maintain certain traits, right? That could, by cross-pollinating, by mixing up genes from different organisms, you're more likely to get different alleles and different traits within a population. And if you're trying to maintain certain traits, that could be an issue. Now, when we talk about cross-pollination, there are two main ways. Majority, well, shouldn't say a majority, but many of our plants are insect pollinated, and that's common for our plants that have white or bright colored flowers that are trying to attract those insects. And then we also have plants that are wind pollinated. Those would be our grasses and our cereal grain crops. Our insect pollinated crops are more of our fruit crops and vegetables, legumes. Now, with our insect pollinated, bees are the most important, right? They are the most important insect in terms of pollinating flowers and pollinating our crop species. However, other organisms are also important. Other insects are also important. Flies will pollinate stuff. Bees will pollinate stuff. And then I'm going to show you some other types of pollinators that I think are just interesting. So bats are good pollinators. They pollinate over 300 species of fruit, such as mangoes, guava, and agave. There are snails that act as pollinators, and I'm kind of going to go over this fast. I just kind of want you guys to see this because I think it's interesting. The world's largest pollinator is this guy right there. That's the black and white ruffed lemur, and they pollinate this traveler's palm. This traveler's palm has these bracts, which you see here, which are leaves, modified leaves around flowers. And this lemur is the only organism that's able to open up those bracts and stick its tongue in to get at the nectar, and it gets pollen on its face, and it moves from flower to flower, and it's able to pollinate this traveler's palm. Again, fun fact, it's the world's largest pollinator. Besides insects and mammals, we can also have reptiles that will pollinate. So here is a gecko that pollinates plants on its island home of the Mauritius. I think that's how you say that. It's pretty. I thought it was a pretty picture. And then my favorite is the possum. So in Brazil, possums will pollinate a weird fungus-like plant that you see here. Similar to the traveler's palm, this plant has these bracts, these modified leaves that cover small flowers, and the possum is the only animal in the area that has nimble enough fingers to move the bracts and get at the flowers and hence pollinate this plant. So one of my favorites. I love possums. I love their hands. They're actually really great benefits for us. They eat ticks, so you should appreciate possums. Okay. So kind of interesting aside on some pollinators. So our plants, because cross-pollination is so important in wild plants and wild populations and all the benefits of that genetic diversity, some of our plants have developed adaptations to facilitate it. And they have morphological adaptations. So some of our plants are dioecious, and this means that they have male and female flowers on separate plants. So you have separate male plants and female plants. This is true for asparagus, holly trees, pistachio. On the other hand, you can have monoecious plants where they have separate male and female flowers, but they are found on the same plant. So this also facilitates cross-pollination, maybe not as much as with dioecious, because in our monoecious plants, some self-pollination is possible. But we see this in our cucurbits, corn, walnut, pecans, oak. They are all monoecious plants. Besides having different flowers on different plants or different male and female flowers, we could have the development of male and female parts occur differently in the flower. That's what you see here with dichogamy. So maybe the male reproductive part forms first and sheds its pollen before the pistil is receptive. The pistil, which is the female reproductive part, is receptive to it. And that is going to essentially improve the odds of cross-pollination. And that is true in our carnations. And then we can also have just the different arrangement of flower parts. So our pistils could be very tall, our stamens could be very short, and that's going to prevent, as you see right here, and that's going to help prevent pollination, self-pollination, right? It's unlikely that the pollen grains from here are going to get up top to the top of the pistil up there. Besides morphological mechanisms. We also have genetic mechanisms. Some of our plants are self-incompatible. Our Wisconsin fast plants that you guys are going to plant in the lab are self-incompatible. So with this, this is controlled by genetic mechanisms. There's a gene that these plants can that these plants use essentially, and they're able to identify a protein that they can recognize as themselves, right? That this is protein to them. And when they recognize that on the pollen, it will abort the growth of the pollen tube. So it will never grow and never get down to the ovaries, and it will never pollinate. It can never pollinate the ovules or fertilize the sperm, won't fertilize the egg. So those are, besides these morphological adaptations, there are genetic mechanisms that are used to prevent self-pollination or self-fertilization and promote cross-pollination. And lastly, I talked about Meiosis, which is a little more rare. This is a condition which so the female reproductive part of the plant is the pistil. It consists of the stigma, which is the part on the top, a style, which is a long tube, and then the ovary. And within the ovary are ovules, and within each ovule is a number of different cells, but importantly the egg cell. And what happens with pollination is that pollen grain lands, it grows a pollen tube, sperm comes down, the pollen tube grows down into the ovary, into an ovule, sperm travels down and fertilizes the egg cell. Well, sometimes, and that's going to form a zygote and form the new embryo. Sometimes that doesn't occur, and instead of that union of the egg and sperm forming the new embryo, a cell within that ovule is going to develop into the new embryo. And that is going to be essentially, it's going to be a clone of the mother plant, right? So instead of having this mixing of genes from potentially separate plants, we just have this mother plant producing seeds that are exactly genetically identical to itself. And that's apomixis. And that we'll see in a little bit, that has a distinct line that is important in some citrus fruits. All right, so we need to know how our plants are pollinated and how they reproduce. And if we have this information, if we are trying to breed plants, we need to know how to control the genetic variability that could occur if there is cross-pollination. Right? We do this simply through isolation, right? We're trying to breed a population. We want to make sure that all the plants in our population are only breeding with each other, right? If they cross-pollinate or if they self-pollinate, breeding with themselves. And we don't want any outside influence, outside plants, outside pollen getting in and potentially mixing in new alleles in our population. So we simply do this through isolation. And this isolation can be either physical isolation or sexual isolation. When I talk about physical isolation, it's really just distance, right? So planting them away from potential other populations or enclosing them in cages, right? And the distance that you need is going to depend on whether our plants are more likely to cross-pollinate or self-pollinate. So with our self-pollinating crops, sometimes you only need to plant them apart or plant separate populations only about 10 feet apart, right? Because there's not much cross-pollination. You don't have to worry about the pollen from different populations interfering. We can, so 10 feet is at a minimum, up to about 200 feet for some that have a little bit more of cross-pollination. For our insect pollinated or wind pollinated plants, we need a much greater distance, right? Because with them, we have to make sure that these insects or wind is not going to carry pollen from one population to the other, and so we need to space them further apart. So for insects pollinated plants, the minimum distance could be anywhere from 1/4 of a mile to a mile, depending on the species. And same with wind pollinated, anywhere from 1/8 of a mile up to 2 miles, that you need to separate populations if you're trying to keep them isolated and prevent the influx of new genes from other populations or wild populations. That's physical isolation. You can also isolate them sexually, and we do this by pollen control. So one of the ways we can do that is we can emasculate the flowers, so we can remove the stamens, that male reproductive part that produces the pollen. We can remove them from our flowers and then manually pollinate our flowers. So you guys are going to manually pollinate your flowers, you're not going to emasculate them. You don't have to do that. You can, if you have dioecious plants, and these are plants that have male plants and female plants, so all male flowers and all female flowers, you can separate them. So as an example, cucumbers, the female plants are preferred for the vegetable crops. So you can plant primarily a field of female plants and then add in a couple males. And then before you get to fruit production, you rogue the males out. So you discard the males. And so it's one way you can use pollen control and control pollination. Or you can use something like that, which are pollination bags, right? You can just isolate individual flowers to prevent outside pollen from coming in. Okay, so that's a little bit, we looked at some of the genetics of breeding. We looked at some more practical applications, so knowing how they pollinate and some of the ways we can control pollination, really, isolating populations, whether physically or sexually. And now, lastly, we're going to end by looking at how people both historically and currently manage seed populations, right? And what we're essentially going to be looking at is we're going to start with least managed and go to more managed, right? So historically, crop improvements came from managing and refining wild populations. And this is still done in forestry, in the nursery industry, and in native plant propagation. So this is important in some of the stuff that I do for my day job in habitat restoration. We utilize wild populations. We get seeds from wild populations. So what this is based on is that in nature, seedling populations have evolved over time to be adapted to the environment in which they're found at particular locations, right? We refer to this as like an ecotype. So essentially you can have a plant that has a huge range, maybe across the United States, but in each local region, it gets local adaptations to that climate or the particular environmental characteristics there. So sometimes historically we could have gone around and seen that, hey, maybe over in New Jersey, this plant has this particular adaptation, these particular genes that could be really beneficial, and we could take that and breed that in with other plants, right? So we could have done that by looking at these wild populations. Or in the case what I do, if we are trying to restore an area and we're going to use wild plants to restore it, I want to collect plants or seeds from plants from an area, from plants that are grown nearby, because I know that they are going to be adapted to that condition. So if they are more likely to survive and prosper if I use plants that are well adapted to New Jersey than if I use plants that have grown and adapted to somewhere in California, just say, right? And we refer to this as provenance, and that's just the origin of where the seed is. And so I use Spartina patens, which is a salt marsh grass. There is a variety, Avalon, which is was taken from Avalon, New Jersey. So that's a good variety. I know if I'm doing wetland restoration in coastal New Jersey, I'm going to want to use that variety as opposed to a different variety that is potentially adapted to some other environment. So historically, we could have managed some wild populations, looked for crop improvements from these local adaptations. Along the same lines, instead of looking at wild populations, we have what are called land races. And these are seed populations that have been maintained by farmers for generations, right? So they're growing their crops. Each year, they save a portion of their crop that is going to be used to produce the next cycle. Now, the land races can be variable. So they're not so much concerned with all of the crops breeding true, so producing the exact same traits. There may be some variation in yield, in fruit size, etc. in your land race, but they're going to be very well adapted to where you're growing them. A great example is cannabis production. So there are a number of land races that people have grown for years in different areas, and they have different names. But they essentially are grown in an area, and they're very well adapted to those local conditions. Again, they may not breed true. They may not all have the same traits. The offspring may not all have the same traits as the parents, but they're going to generally grow really well where they're found, right? So this is more common historically. Now land races have largely been replaced by modern cultivars, but historically they were more important. We also have heirloom varieties, which is very similar to land races. Our heirloom varieties are vegetable and flower varieties that have been maintained just like our land races by saving seeds from the previous year's crop, typically by families. Our heirlooms are a little bit more managed, I'd say, than land races. Typically, your heirlooms have been bred to have particular traits, and those traits, by and large, will be passed down. They will be the same amongst new generations. So there are Seed Savers Exchange is a pretty well-known exchange where they maintain heirloom varieties. They sell seeds. Southern Exposure does as well. I like both of them. I've bought seeds from both. And like I said, with our land races, a lot of heirloom varieties have been replaced nowadays by modern cultivars, which tend to be more uniform and high yielding, right? Our heirlooms are, when you grow your heirloom varieties, they may not be as uniform. Again, the genetics, there may be a little bit variation in there. They may not be as high yielding as our modern varieties. I like, for just gardening that I do, I like heirloom varieties. I think it's interesting. I like growing the different, I think it's fun to grow older varieties of stuff. There's like a historical background with heirloom varieties, so I enjoy it. But again, the downsides is that, or at least why they've been replaced, is because our modern cultivars generally are much more high yielding than our heirloom varieties and uniform. Which brings us to our modern day cultivars. So a cultivar is a plant population that shows a minimum of variation and that can be propagated true to type for at least one characteristic and is unique compared to wild species or other cultivars, right? So generally, at least for one characteristic, typically more, our cultivars are going to breed true to type. So if we breed these together, the offspring are going to have the exact same traits, right? And what this means, if we're thinking about genetics, is that they are going to be very homozygous for that trait, right? Whatever that trait is, or whether they're multiple traits, they're going to have the same allele. So if we cross them together, the offspring are going to have the same alleles. And cultivar is often used interchangeable with variety. I don't know if there's a legit difference between them. Cultivar is just like short for cultivated variety. There may be some like specific there, but typically people will say cultivar, they'll say variety, and they use those terms interchangeably. So we also have a couple categories of seed propagated cultivars that you should be aware of, right? And this is now the most managed, right? When we went from wild populations to land race to heirloom, our cultivars are generally pretty highly managed. They've been bred to be specific, and we maintain that. So our line is a seedling population whose genotype is maintained relatively intact during consecutive generations, right? And we maintain this largely by self-pollination, if they are capable of self-pollination, or that breeding management that we looked at, right? Isolation, whether physical isolation or sexual isolation. Now, there are different types of lines. I'm going to go through them. We don't need to know them specifically, but you should know the general idea of a line. So a pure line is created from the descendants of a single parent, and it typically takes around 6 to 10 generations to produce a true breeding, homogeneous, and homozygous cultivar line. And I don't know if I mentioned homogeneous before, but homogeneous means that all the seedlings are going to look similar. They're all going to phenotypically similar. They're going to have the same traits, right? So homogeneous goes with homozygous. When we have all the same genes, it's going to produce all the same traits generally. So, and that's going to take, if we're breeding a line, about 6 to 10 generations. Now, each generation, if we are breeders, we're going to eliminate or rogue off plants or any plants that don't have the traits that we want. And we just do this through time and time and time, and eventually we should get a population that has our preferred traits. Now, an inbred line is created through enforced self-pollination. So if we can pollinate, we will, if the plants can self-pollinate, we can create inbred lines where we're just continually self-pollinating them. And this is going to result, again, in a homogeneous and homozygous line. And we use inbred lines to create hybrid plants. Now, hybrid plants are very important when we're talking about corn, which is why I'm going to bring it up, because corn is so abundant, such a important crop species. Almost all the corn that people plant is hybrid corn, unless you're doing heirloom corn in a garden. Commercial corn production is going to be almost hybrid corn, and it's because of this trait, which we refer to as hybrid vigor. Hybrid vigor or heterosis, and this simply means that hybrids have improved function or increased function over their parents. And hybrids are essentially the offspring of two inbred lines, right? And you can see this improved function, right? Here's hybrid corn in the middle, here are the parents on the side, right? Offspring have much larger. Look at the size of the corn cob, right? Big difference with our hybrid corn. Better yields, typically better stress tolerance. Like I said, almost all corn that is planted is hybrid corn. And again, if we're thinking about the genetics of it, we are combining inbred lines. So those inbred lines are going to be homozygous, right? Each of them are going to be homozygous, and then we breed them together to create a heterozygote, and that's going to be our hybrid corn, right? So here's a parent genotype, AA, little a, little a. Here's another parent, big A, big A. All of the offspring are going to be heterozygous, and these are our hybrids. Now, the downside of hybrid corn is that you can't save that corn, some of that corn, and replant it the next year. Because we know from our what we looked at the way beginning of this lecture, if we cross two heterozygotes, they do not breed true, right? Only 50% of the offspring will have the same genotype. The rest will differ. So if you save your hybrid corn, you plant it again, you're not going to get the same yields that you would if you just planted hybrid corn again. So that is the downside. But hybrid corn is important. Again, because like you see here, the improved yields that you will see, and stress tolerance. So that is a line. Those are inbred lines. Those are how inbred lines can be used to produce hybrid corn or hybrid plants. We also have things referred to as multilines, which is a mixture of different pure lines that we breed together. So we have two separate lines that are going down. They're all very homozygous for their traits, and then we breed them together. And the reason we may do this is to create a little bit more genetic variability, because we said, again, the importance of genetic variability is it creates or provides material, provides genetic material that allows these populations to be more resilient to diseases or environmental change that can occur, right? Without that genetic material, they can't adapt as quickly. They may not have the genes that could deal with these different stressors that they may be under. So sometimes we're going to breed lines together to give them a little more genetic variability. And these are multi-lines, and there are different types of multi-lines. You don't need to know specifically the difference, but there can be synthetic lines and composite lines. The difference is whether they're combining inbred lines or not. Again, don't need to know. The importance is the idea behind multi-lines is to increase a little bit of genetic diversity. So those are lines, and then we also have what are referred to as clonal seed cultivars, and this is where that apomixis comes back into play. So again, that apomixis is where sometimes within the developing ovule, which is going to turn into the seed, instead of the union of the sperm and egg becoming the embryo of the plant, just a regular plant cell will become the embryo. So it creates a clone. So these are automatically going to have the same genes as the parent plant. Makes it real easy. And this is important for some of our citrus plants as well as mango and apple. They can produce these types of seeds, and then we know they're going to have the exact same genes as the parent plants. Kind of helps. We don't have to worry so much then about managing or isolation for pollination, right? So those are clonal seed cultivars. The last thing we're going to talk about is legal protection of cultivars. So there is the Plant Variety Protection Act, which extends patent protection to seed propagated cultivars that can be maintained as lines, including hybrids. A new cultivar must be novel, distinctive, and stable, so their characteristics must remain unchanged during seed production. and your patent lasts for 20 years for most plants, 25 years for trees and shrubs and vines. So if you guys get into this and you want to create your own line, your own cultivar, you can do that, and you can get legal protection, and you can then sell your cultivar to different people, and they'll have to pay you money. So something to think about. And with that, that is the end of the lecture. Hope you guys enjoyed, and I will see you guys next week.